Assalamu alaikum. Dear professor, dear colleague, I'm very happy to be here today, and I am very honored to have this opportunity to be here. A particular thank to Dr. Hala Gabru for giving me this opportunity. Today I'm going to speak about the evaluation of the potential of the adipocyte stem cells differentiation into cardiomyocyte-like cells in vitro. I'm Dr. Eman Ahmadi. I'm a lecturer of clinical pathology, a consultant of hematology, a certified professional trainer in the International Board of Certified Trainer. I had a professional diploma in quality hospital management and in infection control and safety. Stem cells are unspecialized cells with the ability to renew themselves for a very long period of time without significant changes in their general properties. They can differentiate into various specialized cell types under certain physiological or experimental conditions. What are the types of stem cells? There is the embryonic stem cells, the fetal stem cells, adult stem cells, amniotic stem cells, adipose tissue-derived stem cells. The MSCs are also called mesenchymal stromal cells. They are subset of non-hematopoietic adult stem cells that originate from the mesoderm. They possess self-renewal ability and multilineage differentiation. MSCs exist in almost all tissues. They can be easily isolated from the bone marrow, from the adipocyte tissue, the umbilical cord, fetal liver, muscle, lung, and can be successfully expanded in vitro. Cell therapy for cardiac regeneration. Cardiovascular disease is the main cause of mortality worldwide. Different strategies have been tested in order to recover the loss of the working myocardium, such as the delivery of the growth factors, cytokines, in vivo reprogramming of the myofibroblasts, and dividing non-cardiac cells into the cardiomyocytes or cell therapy. Adipose tissue can be easily obtained in large amounts by means of simple liposuction procedure. So it can be used as a novel, promising alternative source of stem cell for cardiovascular repair. This study was performed in Munwafiyah University Hospital with my colleague Dr. Asma Ishami and my professor Dr. Siham Khudir. Actually, the aim of our work was to study the ability of the adipocyte-derived stem cells to differentiate into cardiomyocyte using 5-azacitidine. The subjects and methods. The mesenchymal stem cell isolation from the adipose tissue. Stem cells were isolated from human adipose tissue, obtained from liposuction samples, and then they were identified by their morphology and their surface markers. The mesenchymal stem cells were subcultured in differentiating media, which contained 5-azacitidine as a cardiomyocyte differentiation factor. So they'll be differentiated into cardiomyocyte. I will detect them by different methods. The first one is the morphology of the cardiomyocyte, the flow cytometry cardiac markers. The markers are used where cardiac troponin, connexin 43, and vimentin, and immunofluorescence staining for the cardiac alpha tropomyosin detection. So how I can isolate the stem cells from the adipose tissue? Actually, this work was done in collaboration with the plastic surgery department. They give to me the lipoaspirate. The lipoaspirate washing, if I cannot work lipoaspirate immediately, and I had to leave it for a certain period of time, never put the lipoaspirate in the refrigerator, because the refrigerating process will affect the digestion. So if you had to leave the lipoaspirate for a certain time, you better leave it at room temperature. Okay, so the lipoaspirate washing, I will perform the wash using the HBSS, okay, with antibiotics and fungizone. I will perform a vigorous shaking. So I will perform a very vigorous shaking for 5 to 10 seconds. I will repeat the shaking for 3 times or several times until it is completely clear. As you can see, there is the lipoaspirate, which is obtained from the surgical department. Okay? Then I will leave it to settle on the bench for 5 minutes. When I will leave it to settle on the bench, okay. I will leave it to settle on the bench for 5 minutes. Then the adipose layer will float on the surface. okay? And the remnants will stay on the bottom. So after several wash, I will finally obtain this. And after several shaking, I will obtain this. When I obtain this, that means that the adipose tissue is completely clear, and then I can work on it, the digestion process. So, the digestion process. Actually, I had tried a lot of digestive enzymes and a lot of methods. The first method I have used is a mechanical method. Actually, it doesn't give to me any kind of results. I have started the trypsin, and actually, it was not successful with me because I thought that it was cheaper than the collagenase. But finally, I go to the collagenase type A, and collagenase A type 1, and do not lose your time trying different methods of separation. Just go to the point and use collagenase. It just had to be freshly prepared in order to work very well. So the aim is, I will put collagenase, okay, on the which will be freshly prepared on HBSS, and then I will put it on the lipoaspirate. Then I will put it on the incubator for 1 hour, okay, for 1 to 2 hours on a shaker. After this, when there is complete digestion, after this I will work on the separation of the stromal vascular fraction. After the collagenase direction, many processes or many things will be performed. The first one is the collagenase digestion. Then I will separate the stromal vascular fraction. Then I will separate the stromal stem cell from the stromal vascular fraction. Then I will remove the erythrocyte by the ammonium chloride. I will remove the cell clumps on the remaining undigested tissue. Then I will culture the stromal stem cell in a media, Daman media, which contain fetal bovine serum at a concentration of 10%. Then I will trypsinize for the gathering of the stromal cells, and then I can identify the adipocyte-derived stem cells. So as we can see that, after I use collagenase, it will form the adipose tissue will form a homogeneous soup-like, it will be like a soup-like solution. And then I will put it on the centrifuge in order to obtain this infranatant, or this, which is a stromal vascular fracture, okay? This part contains the stem cell which I need. I will repeat this several times until it is completely clean, and then I will put on it ammonium chloride in order to get rid of the RBCs. Then I had after several wash, I had a purified concentrate of the stromal cells or the stem cells, which is obtained from the lipoaspirate section. Then I'll be ready to culture these stromal cells on the different plates on the media, which is a Daman media, as you can see in the tissue culture flask. After this, I will trypsinize the cell, and then I will begin the differentiation process. The cardiomyocyte differentiation, it will be by using 5-azacitidine at a concentration of 9 micromole per liter for 7 days. And then I will evaluate the cardiomyocyte differentiation by post-induction morphological changes, by identification of the cardiomyocyte by specific cardiac cell surface marker like cardiac troponin, connexin 43, and vimentin, and finally by immunofluorescence staining, which is cardiac alpha-tropomyosin immunofluorescence detection.